next session with Dr. Chandrashekhar, where he will be diving deeper into the topic of fecal transplant. So, Dr. Chandrashekhar, we already got some information from what you're doing, but I'm sure we'll have next 30 minutes to dive deeper into it. There are many questions in the chat also regarding the gut microbiome, regarding the probiotics. I'm sure some of them will be answered in your presentation, and a couple of them will take it once you finish. Over to you, sir.
Yeah, thank you for the opportunity. I'll tell you the context of fecal transplant, and it will be different from the world's perspective. So, the kind of hypothesis we are working on, it is like I may be in the confirmation bias, but it looks like most opt for the autism, because it is a whole body disorder. The concept we are working may give a lot of conclusions about the disease. So I'll start my presentation. So this is our clinical trial. It is called gut cement restoration therapy. It is in the phase 1 first now, and its public title is Fecal Microbiota Transfer Therapy for Autism. The entire details of the investigation is in the public domain in the CTRI website. So if you go into the ICMR CTRI website, CTRI registry, and type CTRI /2022/02 and /040557, or Fecal Microbiota Transfer Therapy for Autism public title, you will be able to get the entire details of the trial. And in this trial, we are working on epigenetic signatures and gut microbiome profiles in the autism spectrum disorder. And we will be assessing the clinical outcomes of fecal transplant. And also, we will be assessing the causal association between the gut microbiome and the development of the ASD. And let me tell you about the fecal transplant first. Fecal transplant is a procedure to collect the stool or feces from the healthy donor and introduce that stool into the patient's gastrointestinal tract. This modality is actually approved for the ulcerative colitis and Clostridium difficile infection. And this is not yet approved for the autism. And we are investigating this modality, and we are seeing good result with kids with GI symptoms and food allergies. And the rest of the children had minimal response, and we didn't consider it may or may not be because of the fecal transplant. So the hypothesis we are working on is like there are certain microbes. There are 18 tags of microbes that are residing in the mother uterus. So that are vertically transferred to the fetal colon. So during the embryonic development, the genes of these microbes and fetal genes act as a common gene pool. And the gene-to-gene interactions in this common gene pool makes the epigenetic medium. When this epigenetic medium is necessary for the entire structure development and its function of the human body. So when these microbes actually act as a pioneering microbes, and they develop human compatible ecosystem. These are the microbes which will not allow pathogenic microbes there, and helps in development of the compatible ecosystem. When these microbes are absent, so our genes are like losing their effects. Like there is some sort of reduction in the genetic component. So the epigenetic medium gets affected. So the cord-blood toxins that are entering into the fetus that are preventing this evolutionary related 18 tags of microbes that reside in the uterus in and they prevent their engraftment, and they prevent development of the human compatible ecosystem. Because of these toxins, as these pioneering microbes are not developed properly, the microbes that are capable of surviving the toxic environment become the pioneering microbes and aid in the phylogenetically different ecosystem. So this is the actually challenging part in the autism. When whether we are giving some probiotics or medicine, something, if we are giving this is the probiotic, this is the ecosystem that imposes challenges for us. Like they will be responding differently compared to the normal individuals for any of the medicine, any of the probiotics, anything. So this is the concept we are working on. Fecal transplant is also, we are doing fecal transplant for replenishing those important microbes which are required for the correction of the epigenetic medium. So I will talk about the cross epigenetic. Because of there is a disruption in the gene pool. The epigenetic medium is disrupted. Because of that, there are certain mechanisms that are going abnormal during neurodevelopment. So recent postmortem studies of the brain tissue studies, they have identified 584 genes are overexpressed and 558 genes are underexpressed. And there is a differential alternative splicing events happening in the 833 genes in the ASD cortex when compared to the normal individuals. So there is like it is like a gross epigenetic errors. And you can see what is the differential. It is underexpression and overexpression is like if reelin, I'll give you one example of reelin. Reelin is a gene that is required for the signaling of the lamination of the brain. So that is the reelin gene is underexpressed in ASD, and the levels of the reelin signaling molecule are 40% compared to the normal individuals. Because of that, there is a lamination defects in the neurodevelopment. And you can see there are certain proteins that are overexpressed. One of such protein is ALF4. That actually does microglial inactivation. Microglia are the responsible for the functional circuitry formation. When they go inactive, the functional circuitry formation affects. So this is like one example of overexpression, one example of underexpression. But if there are more than 1,000 genes undergoing this process, you can understand how many mechanisms are affected. And differential alternative splicing events. You can see this picture. In this picture, this is like this RNA is a primary transcript from the DNA. It has both coding and non-coding regions. So coding regions are called exons and non-coding regions are called introns. So there is a mechanism called splicing. So this is a normal alternative splicing. Alternative splicing means cutting. It is like they cleave this mechanism cleaves the non-coding parts and rejoin the coding parts in a certain fashion to produce a protein. So we have around 25,000 genes, but we have 5 lakhs more than 5 lakhs proteins in our human body. So it is one gene, one protein hypothesis no more is obsolete now. This mechanism is affected. So if a protein A has to be formed, like the exon 1, exon 2, exon 3, exon 4, exon 5, it is joined in a particular fashion. So this happens in normal individuals as well. But in autistic individuals, this protein formation gets affected. Like there are like events happening like mutually exclusive exons and retained introns and so many other different types of splicing errors, making some brain proteins as isoforms. If this protein A is the desired protein, instead of this desired protein B or protein C are formed. In this, you can see the exon 3 is missing. In this, you can see exon 4 is missing. Like this, sometimes they may jumble up and the protein formed becomes dysfunctional. So this is how the mechanisms are affected. So in this picture, you can see this is the neural tube, proximal part of the neural tube. This is the early, the autism starts in the early phase of neurodevelopment itself. In this, we can see this is the ventricular zone. ventricular zone is the stem cell producing area. The first formed cells from the ventricular zone form a ladder-like cells. These are called radial glial cells. The next formed cells subsequently form neuronal stem cells, travel on these radial glial cells, and form 6-layered structure in the neocortex, 5-layered structure in the archicortex, and 4-layered structure in the paleocortex. So this mechanism is called lamination. So this mechanism of traveling neurons at the radial glial cells is called neuronal migration. So this neuronal migration requires a signaling protein called reelin. Reelin is very much important for the neural development. So this is only 40% present in the autism. So because of that, neural migration is affected. When the neuronal migration is affected, so I can show you this picture. So this is the ventricular zone, and there is a circle of tissue that did not migrate to the cortex, and they stay back in. These kind of errors are called heterotopias. So you can see once after forming the lamination, the precursor cells have this are like this, small and less branched, and they undergo neuronal differentiation to become long neurons with highly branched, and they connect randomly with the other adjacent neurons and make so many connections. Then microglia comes into the picture and to read the missense synapses. And the missense synapses are read by the microglia, and they cleave the unnecessary synapses and forms the functional circuitry. This is the mechanism generally in normal neural development. This neuronal differentiation is also affected, and the cells will not reach their full potential. And most of the cells are they under their differentials alternate. This differentiation is affected, and they do not grow further. That is the reason we have the more number of neurons are present. And when this neuronal migration is affected and lamination is affected, there is a negative feedback to the ventricular zone. When there is a negative feedback to the ventricular zone, that the brain is not developing, then the ventricular zone starts producing more number of stem cells. This we can see from the autopsy studies. This is called subependymal nodular dysplasia. This is like this is a germinal layer. There are at least 100 times more number of stem cells are there in autistic brain compared to the normal, because the brain is not developing. So there is a negative feedback to the ventricular zone that brain is not developing. The ventricular zone starts producing more and more stem cells. This is called accelerated neurogenesis, and that's why there are 67% of extra number of neurons compared to the normal individuals. That's why the density of the autistic brain is more than the normal child. So these are the and these neuronal differentiation defects are mainly observed in these colored areas. You can see, and there are like social impairments. The social impairments are because of the this orbitofrontal cortex and anterior cingulate gyrus, fusiform gyrus, and superior temporal sulcus, and amygdala mirror neurons, inferior frontal gyrus, and posterior prefrontal parietal cortex. These are the important areas that are underdeveloped in autism that cause social impairment. And likewise, communication deficits like inferior frontal gyrus. If the Broca's area is not developed, they will not be able to speak. And the language areas are affected. They cannot be. They will not be able to speak. They will not be able to process the language. So we have seen certain kids. They have Broca's areas developed, but their language areas are not developed. When the language areas are not developed, they don't comprehend the language, and they just do a parrot kind of speech and like echolalia and all. So the superior temporal sulcus, basal ganglia, substantia nigra, thalamus are major things for the communication deficits. There are some of the areas like orbitofrontal cortex and anterior cingulate, basal ganglia, and thalamus for the repetitive behaviors. In autism, these are the areas they are underdeveloped, and the gray part of the brain area, though it is developed, they have some sort of functional dysfunction because of the epigenetic errors. And we are targeting by doing FMTs, do a proper bowel cleanse to reduce the microbial overload, and you have to suppress the phylogenetically different ecosystem, and then do fecal transplant on a daily basis for few weeks. So which was the Adam study was there for 7 weeks, and probably they're changing it to 16 weeks in the phase 2. So we have done from 10 ranging from 10 sessions to 16 weeks. So when the children getting longer sessions on a daily basis, and so FMT and a clean colon are responding faster. So I'll tell you about the outcomes of the fecal transplant. So I try to depict in this one. So if the autism, so we have seen that from the autopsy studies that epigenetic errors are there. So epigenetic errors can be because of the fetal genes itself. When there is good microbiome in place, but still there is problem with the genetics. So in such conditions, there are certain epigenetic errors are happening. In that case, doing fecal transplant will not help, and it is irreversible. And we can see the cases with the gene environment interaction. Like I said, if they have phylogenetically different ecosystem, that can be reversible. And we have seen the younger kids are the fast responders, like less than 3. We have seen kids responding faster less than 3, and 3 to 7 there there is a moderate response. After 7, it is the response is very slow. And so you can see with the epigenetic errors, there are certain structural changes that we depicted earlier in this one. There are functional changes also there. Functional changes are these are ongoing epigenetics. Functional changes when you touch a surface, when you taste something, when you see something. So that signal generally has to go through the sense organs to the brain, and this process happens with the formation of neurotransmitters. Every stimulus has a custom-made transmitter composition, and there is a lot of protein synthesis activity going on during this sensory inputs. So whenever we touch a surface or see something, that transmitted through there is a production of neurotransmitters. There are 5 lakhs protein neurotransmitters produced per minute at every synapse. So you can imagine how much of epigenetics is involved here. So when they have a different phylogenetical ecosystem, they have there is epigenetics affected here. So they produce there are some gene expressions happening, and neurotransmitter composition is getting affected. And this is one of the reason we have seen they can they have some have bland taste, and they don't mind eating 10-15 lemons at a time. So these are the because they are the stimulus is not producing such kind of signal. So the neurotransmitters are affected. So that's why there are certain sensory issues. Those kind of sensory issues like visual stimming and touch kind of sensory issues and all, these were settling after fecal transplant. And the structural deformities that formed during the wound, like those areas, these colored areas are developing very slow, and these kind of when the neurological deficits, though the neurological deficits are there, the child is becoming comfortable because this gray part of the developed area is becoming functional after fecal transplant. So I'll tell you about some updates from our pilot studies and our trial. So we if we do a fecal transplant with the child's stool inside the colon, they generally show some symptoms of aggression and all. Just because I'll tell you about one animal study. In that animal study, they have taken autistic child poop to their autistic child stool and transplanted it into the pair of germ-free mice. Those pair of germ-free mice developed the behaviors, autism behaviors, and their offspring developed autism. and their brain tissue studies showed that the pattern of neuronal migration, neuronal differentiation that is seen in the human autism, that was similar in the mice offspring. So it is the stool is one of the causal factor in gut kids. So if we do fecal transplant with that stool in their colon, so the saline mixed in the solution and all that will take all the toxins from the child's poop, and when that enters into the circulation, there is an aggravation of the autism symptoms. So we started doing full bowel cleanses. It is like we started doing fecal transplants and clean colon. Fecal transplants or clean colon are the successful, and if the fecal transplant is done with half bowel cleanse, it is not going to be successful. And one more thing, like it is the phylogenetically different ecosystem they have developed. It is very strong, and it's already does immune priming in our body. And when we do a fecal transplant, that it is not easy to overpower the existing ecosystem. So that is the reason. So it is taking too long course to change the entire gene pool. So and we have seen like some kids, we have seen a few kids like the mother had cholestasis or rheumatoid arthritis during pregnancy, and the child developed autism. Those kind of cases didn't have gut symptoms and all, but they did not respond to the fecal transplant. And there are at least 30% of cases didn't respond, and most of the kids without GI issues didn't respond, and kids with genetic involvement also did not respond. And about the diet. During FMTs, if somebody is taking sugar and junk food, the success of FMT was getting affected. And we have seen increased craving for the sugars after fecal transplant. This may be because of the die-off reaction. And we stopped everybody from eating sugars and junk food and toxin containing, pesticide containing food. And we are trying to keep them on the organic diet. And the symptoms, there are certain symptoms we have seen improved in the first few weeks of the fecal transplant. It is like their gut issues getting settled. This is expected at any age, and their sleep and mood are improving, and their behavior is also improving, like self-injurious behaviors and cranky behaviors. They are improving, and the sensory issues and fine motors slowly improving over a period of 3 to 6 months. And their understanding and cognition are improving in first few days of the fecal transplant, and they are responding faster, better in the therapies also as well. But we did not notice any full recovery till now in these 40 kids. Like one of the kid was diagnosed at 20 months, and we did fecal transplant for that kid at the 20 months age. And it's been 18 months, but we have there is a lot of improvement in that child, and she's almost near normal. But we didn't notice full recovery in that kid as well. So that means once the child develops autism, I don't think we can completely recover the child. So it has to be prevented before it happens. So some of the investigations I'll tell you, like earlier I have told you, we have, after doing fecal transplant, there is changes in the microbiome profiles and the changes in the microRNA profiles. These microRNA actually, they have capacity to switch on and switch off the genes and regulatory aspect, gene regulation. So if the microRNA repertoire is affected with the microbiome, that means there must be some kind of which is human compatible and human incompatible microbiome. So we have to look further studies to understand what kind of microbes are required. And we have also seen like there are certain viruses, bacteriophages in the gut microbiome. They alter their behaviors. Like we have seen some studies like one of the bacteria like Luminococcus, they act differently in one ecosystem, and they'll be doing some other thing in the another ecosystem. So the entire ecosystem might be the we should be targeting. And we have also seen like that presence or absence of certain microbes in the microbiome studies. that analysis doesn't seem to give something to us in the future, because we're only seeing looking at the microbes, this bacteria and all, but there are many other things are there, like viruses, bacteriophages, and so many other things are there in that one that will alter the entire microbial function and whatever their activity. So we are doing further much more deeper research for proof of concept. Probably we'll come up with some solutions in next 2 to 3 years. Thank you.
Thank you so much, sir. This was fecal transplant is definitely one topic where much information isn't available. So having you on this in this conference is definitely very, very valuable to us and all the audience out here. I'm sure they have a lot of takeaways from this. A lot of their questions have been answered. We do have 3 minutes for questions, and I'll take them from the Q&A. So you have a question, Dr. Chandrashekhar. So there's a question. You mentioned dysbiosis of nitric oxide pathway. Have you looked at near infrared to boost nitric oxide?
So we did not look at this nitric oxide pathways. I mentioned about nitric oxide synthase adapter protein. It's an adapter protein. It is nitric oxide synthase is involved in so many these things. We looked at the epigenetics. It is like we will be like we were looking at we have taken the blood samples, and we have seen that particular adapter protein. And there is like this NOS AP related genes were there. One gene was recessive and one gene was dominant. The dominant gene that was supposed to produce certain protein, like we have seen that epigenetic pathways. So that particular NOS AP genes epigenetic pathways targetedly we have done. In that we have seen that that particular gene was undergoing differential alternative splicing event. So that makes a different isoform. That means that adapter protein that is produced is a different protein, and it is an isoform. It is not like a regular adapter protein. Because of that, my child was landing up in a nephrotic syndrome. And how it was happening, the later pathways, we don't have technology or something like that to identify that. So right now, what we have seen is like that nitric oxide synthase adapter protein was forming normally after fecal transplant. That means it has direct role in the epigenetics. There comes our entire hypothesis also is different. Like we have seen the epigenetic errors happening in the inside the womb. So if any gut bacteria that has role in epigenetics, that must be present inside the womb itself. So that's why this gene pool concept has come. So we are working on that hypothesis. It is not yet approved or not a proved concept. We are working on that hypothesis. It is like we after doing some research, we understood that this is an assumption from our side, and we are investigating it in the clinical trials. So we are doing meconium studies. That is, meconium is stool from the day one child.
Mm-hmm.
And we'll be doing cord blood also. We'll take the umbilical cord blood and we'll do the study also. That is like trying to understand the environment of the uterus during pregnancy that the child is developing. It is we consider that the child is developing in a toxic medium. So we have to remove the toxins for these susceptible genes to function normally. So in our case, in my child, nitric oxide synthase adapter protein was a susceptible gene for nephrotic syndrome. And she has something like BDNF and so many other heterozygous mutations were there.
Mm-hmm.
They're also at the risk. Those genes are also under risk. So like because that may be the reason the abnormal real information and many other genes. So we have seen like at least 1800 genes were affected during brain development. So we are investigating that.
Okay. All right. Okay. Thank you, sir. There's still some questions. So I'll request you, if you're still available, you can answer them over the Q&A chat. And we will be sharing the contact details of Dr. Chandrashekhar with all of you. So if anybody wants to connect with him, you can do that, after this session. So thank you, Dr. Chandrashekhar, for being on this panel. Thank you so much.
Thank you so much.